I'm Piotr Zimniak. The major interest of my laboratory is aging, the molecular basis of aging. Now, aging is a difficult problem to study for many reasons. One is that it's very complex, but the other is that serious study of aging has begun only about 20 years ago, and therefore many of the basic concepts in the field are not agreed upon. And that causes a lot of confusion in the field. So what is aging? One idea that is widely but by no means universally accepted is that aging is a consequence of two opposing forces. One is essentially physical-chemical wear and tear that happens inevitably and cannot be prevented. The opposing force are genetically controlled, tightly controlled pathways called longevity assurance pathways that are biologically controlled. And that gives us an opening, because normally the lifespan of an organism evolves not to be maximal, but to maximize reproductive success. However, if we learn something about what these mechanisms are and how they are controlled, we can attempt to modify them for our own purposes, and that is to extend human lifespan. Now, it may seem paradoxical that a topic such as aging is really not very well studied yet, given the huge interest in it that was prevalent for a long time throughout history. In fact, much of our culture, of our literature, of our art revolves around aging and around our own mortality. And that just illustrates this urgent desire to change something, to circumvent aging, to postpone mortality, is not really helpful in understanding of that phenomenon. And the sound scientific basis has been established only relatively recently. So how does our own work fit into this picture? We are studying a compound called 4-hydroxynonenal, or 4-HNE for short, this is a product of lipid peroxidation, of oxidation of polyunsaturated fatty acids. And we have found that an excess of this compound shortens lifespan, and a low level of this compound extends lifespan, probably by modulating these longevity assurance processes that I already mentioned. We have some ideas how that works, although they're in initial stages. The more interesting question is, why look into that? There are many other ways to influence aging that are already known. However, they are mostly genetic. One can change aging by influencing the expression of a number of known genes. In contrast, 4-HNE, the compound that we are studying, is accessible pharmacologically. There are compounds available that will modulate the level of 4-HNE. And that makes it a lot easier to influence the complex trait of longevity. So that is what we are trying to do. While we were studying 4-HNE and its effect on aging, we made the serendipitous observation that the compound also influences the amount of fat that an organism accumulates. And that is something that happened in mice, happened in Caenorhabditis elegans, a nematode that is a commonly used model system for many biological processes, and in other simpler organisms. So it seems to be pretty universal. Again, we have some ideas how that works, how 4-HNE can reprogram metabolism in such a way that it discourages the use of metabolic fuel for energy production, and it encourages the use of metabolic fuel for storage, fat accumulation. And we are testing these ideas right now. But again, why is it of interest? Well, the same reason that I mentioned before. 4-HNE can be modulated pharmacologically, and that gives us another way to affect obesity, which is of course a huge health problem. In contemporary biology, a lot of attention is focused on gene expression, regulation of gene expression, the level of gene expression of gene products. In contrast, we are focusing our research on metabolic processes. After all, genes don't affect life directly, they affect the level of metabolites, because life is the fabric of life, consists of chemicals, of metabolites, and the reactions that link them. So we are trying to intervene directly into this fabric of metabolic interactions and achieve our purposes, and both informational and practical purposes in that way.